Continuation of the previous video about recombination and cloning. So, cloning vectors based on viral DNA. Viral vectors in which the gene or genes of interest are incorporated into the genome of a virus. So, because virus infect cells with high efficiency, the cloned gene can be introduced into cells at a significant higher frequency than by simple transformation. Some viral vectors are specialized for producing high levels of protein encoded by the cloned genes, and other viral vectors, such as the bacterial M13 based vectors, are designed to facilitate sequencing and the generation of mutations in cloned genes. Then, cloning vector based on lambda phage. Lambda phage infects bacteria E. coli and replicates by a lytic or lysogenic pathway. Its genome consists of single linear dsDNA of about 48 kb. However, at either end of the molecule is a short 12 nucleotides stretch in which the DNA is single stranded, described as cohesive ends or sticky ends. The lambda cohesive ends are called the cos site, and they play two distinct roles during the lambda infection cycle. First, they allow the linear DNA molecule that is injected into the cell to be circularized. The second role of this cos site is rather different and comes into play after the prophage has excised from the host genome. At this stage, a large number of new lambda DNA molecules are produced by the rolling circle mechanism of replication, in which a continuous DNA strand is rolled off the template molecule. The result is a catenate consisting of a series of linear lambda genomes joined together at the cos site. The role of the cos site is now to act as recognition sequence for an nuclease that cleaves the catenate at the cos site, producing individual lambda genomes. In the lytic pathway, viral genomes are fully expressed. Viral DNA and proteins are quickly produced and packaged into virus particles, leading to the lysis of the host cell and release of virus particles or virions. In the lysogenic pathway, the phage DNA becomes inserted into the host cell genome and can be replicated together with the host cell DNA for many generations, remaining inactive. Certain environmental changes can trigger the expression of this dormant viral DNA, which leads to the formation of progeny virus, and can trigger the expression of this normal dormant viral DNA, which leads to the formation of progeny virus and lysis of the host. Large segment of DNA of lambda phage are not essential for protective infection and can be replaced by foreign DNA, thus making lambda phage an ideal vector. Several major types of cloning vector have been developed by modifying phage lambda. Then replacement lambda vectors. Only DNA molecules from 37 to 52 kilobase pair in length can be stably packaged into lambda particle. The central segment of lambda genome contains genes that are required for the lysogenic cycle but are not essential for lytic cycle. As a result, it can be removed and replaced by foreign DNA fragment. Using this strategy, it is possible to clone foreign DNA up to 23 kb in length, and such vectors are normally used for making genomic DNA libraries. Then insertion lambda vectors used for making cDNA libraries do not require a large insert capacity. Most cDNAs are less than 5 kb long. Design of insertion vectors often involves modification of the lambda genome to permit insertional cloning into the C1 gene. Then cosmids. Cosmids are vectors that are hybrids of lambda phages and plasmids, and other DNA can replicate in the cell like that of a plasmid, or be packaged like that of a phage. However, cosmids can carry DNA inserts about 3 times as large as those carried by lambda itself. So the key is that most of the lambda phage structure has been deleted, but the signal sequences that promote phage head stuffing cos sites remain. This modified structure enables phage heads to be stuffed with almost all donor DNA. Cosmid DNA can be packaged into phage particles by using the in vitro system. The development of cosmid vectors was based on the observation that about 200 base pair DNA sequence in the lambda genome called cos is required for lambda DNA packaging to phage particles during lytic infection. cleavage caused by the lambda terminase protein during phage packaging produces a 12 nucleotide cohesive end at the termini of the linear lambda genome. Recircularization of the lambda genome after bacterial infection is facilitated by base pairing between the complementary cohesive ends. Cosmid cloning vectors with DNA inserts of 30 to 45 kb can be packaged in vivo in vitro into lambda phage particles, provided that the ligated double-stranded DNA contains lambda co-sequences on either side of the inserted DNA. Because the lambda phage head can hold up to 45 kb of DNA, the optimal ligation reaction in closed-mid cloning produces recombinant molecules with co-sequences flanking DNA segments of about 40 kb. E. coli cells infected with the cosmid packaged phage are also able to support autonomous replication of circular cosmid plasmids because a col EY origin of replication and a beta lactamase gene AMP are linked to the cos sequence. Cosmid cloning system, here we can see F plasmid origin of replication and phosmids contain F plasmid origin of replication and a lambda cos site. They are similar to cosmids but have a lower copy number in E. coli. Then cloning vectors based on M13 phage. A very useful vector for cloning DNA. M13 phage is especially useful sequencing this inserted DNA. This filamentous virus contains 6.4 kilobase per single-stranded circle of DNA. M13 enters E. coli through the bacterial sex pilus, a protein appendage that permits the transfer of DNA between bacteria. The single-stranded DNA in a virus particle called the plus strand is replicated through an intermediate circular double-stranded replicative form RF containing plus and negative strands. Plus and minus, I guess. So only the plus strand is packaged into new virus particles. About 1,000 progeny M13 are produced per generation. A striking feature of M13 is that it does not kill its bacterial host. Consequently, large quantities of M13 can be grown and easily harvested. 1 gram from 10 liters of culture fluid. An M13 vector is prepared for cloning by cutting its circular RF DNA at a single site with a restriction enzyme. The cut is made in a polylinker region that contains a series of closely spaced recognition sites for restriction enzymes. Only one of each site is present in the vector. A double-stranded foreign DNA fragment produced by cleavage with the same restriction enzyme is then ligated to the cut vector. The foreign DNA can be inserted in two different orientations because the ends of both DNA molecules are the same. Hence, half the positive or new plus strands packaged into new virus particles will contain one of the strands of the foreign DNA and half will contain the other strand. Infection of E. coli by a single virus particle can yield a large amount of single-stranded M13 DNA containing the same strand of foreign DNA. DNA cloned into M13 can be easily sequenced. M13 vector RF injected into host replication, single-stranded DNA synthesis plus production of single-stranded recombinant DNA using M13. And phage-mid vectors. Next, phage-mid vectors. Phage-mid vectors are plasmids DNA which contain a small segment of a genome of a filamentous phage such as M13, FD or F1. The selected phage sequences contain all the cis-acting elements required for DNA replication and assembly into phage particles. They permit successful cloning of inserts several kilobases long, unlike M13 vectors in which such inserts tend to be unstable. Following transformation of a suitable E. coli strain with recombinant phage-mid, the bacterial cells are superinfected with a normal filamentous helper phage, such as M13F1, which is required to provide the coat protein. Phage particles secreted from the superinfected cells will be a mixture of helper phage and recombinant phage-mids. The mixed single-stranded DNA population can be used directly for DNA sequencing because the primer for initiating DNA strand synthesis is designed to bind specifically to a sequence of the phage-mid vector adjacent to the cloning site. Then bacteriophage P1. Bacteriophage P1 vectors are very similar to lambda vectors, being used being based on a deleted version of a natural phage genome. The capacity of a cloning vector being determined by the size of the deletion and the space within the phage particle. The P1 genome is larger than the lambda genome and the phage particle is bigger. So a P1 vector can clone larger fragments of DNA than a lambda vector up to 125 kilobase pairs using current technology. And cloning vectors for eukaryotes, next topic. Cloning vectors for yeast, Saccharomyces cerevisiae. Saccharomyces cerevisiae has a genome of approximately 2 into 10 raised to 2 base pairs contained in 17 linear chromosomes, and some strains possesses a type of plasmid known as the 2 micron circle. This plasmid has 6,318 base pairs and is present in a copy number of about 50 copies per cell. Then yeast integrative plasmid YIP. YIP vectors lack yeast replication origin and must be integrated into the yeast genome in order to be maintained during cell division. They are normally present at 1 copy per cell and are very stable. It consists of a segment of the E. coli plasmid PBR322 containing a selectable antibiotic resistance gene for ampicillin and a bacterial replication origin that facilitates selection and amplification in E. coli. YIPs integrate YIP or Y1P. Yeast integrated plasmid. So YIP. Integrate by recombination between homologous sequences on the YIP plasmid and the host genome. Then yeast replicating plasmids YRP. YRP or yeast replicating plasmids vectors contain an autonomous replicating sequence ARS of chromosomal origin in addition to the elements found in YAP. YRPs are capable of autonomous replication, are present at 3 to 30 copies per cell. Then yeast episomal plasmids YEP. Yeast episomal plasmid vectors are based on the ARS from the endogenous yeast to mu plasmid, which contains genetic information for its own replication and segregation. They are capable of autonomous replication and are present at 220 to 200 copies per cell. Then yeast centromeric plasmids YCP. YCP vectors contain both an ARS and a yeast centromere. The ARS can be either chromosomal or 2 mu in origin. YCP are normally present at 1 copy per cell, can replicate without integration into a chromosome, and are stably maintained during cell division. Then yeast artificial chromosome or YEC. Colleagues describe the first generation of yeast cloning vectors that were based on artificial chromosome. YACs are used for cloning very large DNA fragments. These vectors are propagated in Saccharomyces cerevisiae rather than E. coli and are based on chromosomes rather than on plasmids or viruses. The essential functional components of chromosomes are centromere, required for distinction of sister chromatids in mitosis and of homologous chromosomes at the first meiotic division. Then telomeres, required for complete replication of linear molecules and for protection of the ends of a chromosome from nuclease attack. An autonomous replicating sequence ARS elements acts as origin of replication and selectable marker to select positively for chromosomal maintenance. Here we could see schematic diagram of a YAC or yeast artificial chromosome cloning vector indicating prokaryotic gene for resistance to ampicillin, prokaryotic replication origin, yeast auxotrophic markers, autonomous replicating sequence, yeast centromere and telomere. Shuttle vectors. Shuttle vectors are plasmids capable of propagating and transferring or shutting genes between two different organisms, one of which is typically a prokaryote and others a eukaryote, which is a yeast. So shuttle vectors must have unique origins of replication for each cell type as well as different markers for selection of transformed host cells harboring the vector. Maximum DNA insert possible with different cloning vectors. M13 plasmid, lambda phage, cosmid, P1 phage, BSc, AC, BSc principle. Cloning vectors in plants. Another topic. Cloning vectors for plants are either plasmid or viral genome based. Plasmid based vector, viral genome based vector. These two types are there. So TI plasmid based vector. TI plasmid present in Agrobacterium tumefensis or A. Tumescens is naturally occurring gram-negative soil bacteria with two common species, Agrobacterium tumescens and Agrobacterium rhizogenes. So these are being considered as natural genetic engineers for their ability to transform plants. A. Tumescens cause crown gall disease when Agrobacterium rhizogenes infect plants, and production of adventitious roots rather than tumor is induced at the site of infection by transferring a defined segment of DNA called tDNA from its tumor inducing TA large sized plasmid of about 200 kb into the nuclear genome of cells in an infected wound on many dicotyledonous plants. tDNA varies in size between 15 and 30 kb depending on the strain. tDNA carry genes for phytohormone, auxin and cytokinin and opines. The overproduction of phytohormones at the site of infection is responsible for the proliferation of wound cells into a gall, a tumor that can harbor a population of bacteria. Unusual compound opines, octopine, nopaline are categorized by agrobacterium. Schematic representation of a TA plasmid. The tDNA is defined by its left and right borders that includes genes for the biosynthesis of auxin, cytokinin, and an opine. These genes are transcribed and translated only in plant cells. Outside of a tDNA region, there is a cluster of VAR genes, a gene that encodes an enzyme for opine catabolism and an origin of DNA replication which permits the plasmid to be stably maintained in a tumescens. The movement of tDNA is mediated by genes in another region of TA plasmid called VAR genes, virulence genes. The virulence genes are not themselves transferred with the tDNA but mediate the transfer. The genes caused tDNA transfer to plant cell is analogous to transfer of plasmids during bacterial conjugation. Following transfer, tDNA get integrated with the host genome. These properties of agrobacterium DNA transfer system are invaluable for developing a powerful vector system for plant transformation. Any genes put in the tDNA region gets transferred to the plant genome. The DNA is inherently stable once in the plant genome since either the border nor the virulence genes are transferred. The TA plasmid of A lysogenes and TA plasmids are very similar. The main difference between being that transfer of the tDNA from an RI plasmid to a plant result not in a crown gall but in a hairy root disease. Then viral vectors. The potential of a new plant virus as cloning vectors has been explored. One major problem with vast majority of plant viruses is that they have RNA genome, not DNA genome. RNA viruses are not useful as potential cloning vectors. Only two classes of DNA viruses are known to infect higher plants: the Coelimovirus (dsDNA) and Gemini virus (ssDNA), and neither is ideally suited for gene cloning. Next is about cloning vectors for animals. For insects, P element, a transposon, is used as a cloning vector in Drosophila. P element at 2.9 kb in length and contain 3 genes flanked by short inverted repeat sequences at either end of the element. The genes code for transposase and the enzyme that carries out the transposition process. The inverted repeats from the recognition sequence that enable the enzyme to identify the two ends of the inserted transposon. The vector is plasmid that carries two P elements, one of which contains the insertion site for the DNA that will be cloned. An insertion of a new DNA into this P element results in disruption of its transposase gene. But the second P element carried by the plasmid has an intact version of the transposase gene that provides transposase enzyme to carry out transposition. And cloning vectors for insects based on viral DNA are not common. However, dsDNA of baculovirus are used as cloning vector for many insects. For mammal, genome of many viruses are used cloning vectors for mammal. First cloning vector for mammalian cell was based on SV40 virus genome. SV40 is a small virus that infect monkey simian. New genome of many viruses such as adenovirus, papillomavirus, which have a relatively high insect capacity, are used as vectors for cloning genes in mammalian cells. At present, retroviruses are the most commonly used vectors. Genome of many viruses are used as cloning vectors for mammal. First cloning vector for mammalian cell was based on SV40 virus genome. SV40 is a small virus that infects monkey, we mentioned earlier. Then selection of transformed cells. Identification of cells containing the vector molecule requires the presence of a suitable marker gene on the vector molecule, whose expression provides a means of identifying cells containing it. Two popularly used marker gene systems are based on antibiotic resistance genes. A host strain is chosen that is sensitive to a particular antibiotic, often ampicillin, tetracycline, or chloramphenicol. The corresponding vector has been engineered to contain a gene that confers resistance to the antibiotic. After transformation, cells are placed on agar containing the antibiotic to rescue cells transformed by the vector. Then beta galactosidase gene complementation. The host cell is a mutant which contains alpha fragment of a beta galactosidase gene but cannot make any functional beta galactosidase. The vector is engineered to contain a different fragment of the beta galactosidase gene. After transformation by the vector, functional complementation occurs, resulting in active beta galactosidase, which can be assayed by acting on a colorless substance, X-gal, 5-bromo, 4-chloro, 3-idolyl beta D-galactopyranoside to make a blue product. Plasmid vector, LAC-Z5 sequence, F dash plasmid, LAC-Z3 prime sequence, beta galactosidase N terminal alpha protein, then beta galactosidase C terminal omega fragment, X gal colorless, X gal blue color. Beta galactosidase activity can be used as an indicator of gene function plasmids that express the N-terminal alpha fragment of beta galactosidase can be used in E. coli strains expressing the C-terminal omega fragment of beta galactosidase on an F plasmid. And recombinant screening, a selective medium enables transformants to be distinguished from non-transformants. The next problem is to determine which one of the transformed colonies comprise cells that may contain recombinant DNA molecules and which contain self-ligated vector molecules. With most cloning vectors, insertion of a DNA fragment into the plasmid destroys the integrity of one of the genes present on the molecule. Recombinants can therefore be identified because of the characteristics coded by the inactive gene is no longer displayed by the host cells called insertional inactivation. So we were talking about insertional inactivation. We mentioned that. So most commonly the recombinant selection is carried out by insertional inactivation of antibiotic resistance genes. In this case, insertion of new DNA fragment insert occurs at the site between or within the gene that confers resistance towards a particular antibiotic. New DNA inserted in BAM-H1 site. Here we can see recombinant. But the original one is normal vector. Insertional activation does not always involve antibiotic resistance genes. For example, in POC8 gene, which codes for part of enzyme beta galactosidase, is used for insertional inactivation. Recombinant involves insertional inactivation of LAC-Z gene can be identified because of their inability to synthesize beta galactosidase. Beta galactosidase coded by LAC-Z gene causes the breakdown of lactose to glucose plus galactose, LAC-Z to a modified LAC-Z gene codes for the alpha peptide portion of beta galactosidase. Introduction of DNA into the host cell, very important step. In bacterial cells, the process of transferring exogenous DNA into cells called a transformation. There are basically two general methods for transforming bacteria. The first is a chemical method utilizing CaCl2 and heat shock to promote DNA entry into cells. A second method is called electroporation. It uses a short pulse of electric charge to facilitate DNA uptake. Two methods are there, transformation methods. First is a chemical method utilizing CaCl2, calcium chloride, and a heat shock to promote DNA entry into cells. The second method is called electroporation. It uses a short pulse of electric charge to facilitate DNA uptake. The chemical method uses bacteria that are incubated with DNA on ice in a solution containing CaCl2, followed by a brief heat shock at 42 degrees Celsius. Exactly why this treatment works is not understood. Possibly CaCl2 caused the DNA to precipitate into the outside of the cells, or perhaps the salt is responsible for some kind of change in the cell wall that improves DNA binding. So first method of transformation is chemical method. Next one is electroporation. Okay. So the chemical method uses bacteria that are incubated with DNA on ice in a solution containing CaCl2, followed by a brief heat shock at 42 degrees Celsius. Exactly why this treatment works is not understood. Possibly CaCl2 causes the DNA to precipitate into the outside of the cells, or perhaps the salt is responsible for some kind of change in the cell wall that improves DNA binding. In case of plant cells, there are two kinds of gene transfer methods, like direct vector-less and indirect vector-mediated gene transfer. Direct gene transfer uses physical, electrical, or chemical means to deliver a gene of interest into the host plant cell. These methods are species and genotype independent in terms of DNA delivery. The examples of direct gene transfer is liposome encapsulation, electroporation of protoplasts, microinjection, particle bombardment, physiochemical uptake of DNA, and others. Physiochemical uptake of DNA. This method is based on the ability of the protoplast to uptake the foreign DNA from the surrounding solution. Isolated plasmid DNA is mixed with the protoplast in the presence of polyethylene glycol (PEG). After 50 to 20 minutes of incubation, the protoplasts are cultured in the presence of appropriate selective agent. Protoplasts are regenerated and putative transgenic plants are further characterized for conformation. Then liposome encapsulation. Wait a minute. Physiochemical uptake of DNA. This method is based on the ability of the protoplast to uptake the foreign DNA from the surrounding solution. Isolated plasmid DNA is mixed with the protoplast in the presence of a polyethylene glycol. After 15 to 20 minutes of incubation, the protoplasts are cultured in the presence of appropriate selective agent. Protoplasts are regenerated and putative transgenic plants are further characterized for conformation. Then liposome encapsulation. Liposome encapsulation procedure was developed to protect a foreign DNA during the transfer process. DNA enclosed in the lipid vesicle penetrates into the protoplasts, where lipase activity of the protoplast dissolves the lipid vesicles, and DNA gets released for integration into the host genome. Electroporation of protoplasts. Electroporation involves the creation of pores in the cell membrane using electrical impulses of a high field strength. Reversible breakdown of the membrane allows the entrance of foreign DNA. Generally, protoplasts are used since they are exposed to plasma membrane. Then microinjection, extensively used in the animal cell. Microinjection of DNA into plant cell has achieved only a limited success. This is largely because of difficulties in getting the protoplasts immobilized and injecting DNA into the protoplast without damaging the tonoplast which surrounds the plant cell vacuole. Then particle bombardment. Particle bombardment of microinjection or microprojectile bombardment or biolistic transformation employs foreign DNA coated high velocity gold or tungsten particles to deliver into plant cells. Different approaches are being used to accelerate the particle. Particle gun accelerated particle penetrates even deep into the tissues. This method is being widely used because of its ability to deliver foreign DNA into regenerable cell tissues or organs, irrespective of the monocoats or dicoats. Because of the physical nature of the process, there is no biological limitation to the actual DNA delivery. This it is genotype independent. Then vector mediated gene transfer. Among the various vectors used in plants transformation, the TA and RI plasmid of trumifensis and rhizogenes respectively has been used extensively. These bacteria are also known as natural genetic engineers of plants, since these bacteria have ability to transfer tDNA of their plasmid into plant genome upon infection of cells at the wound site. TA and RI plasmids can be used as gene vector for delivering useful foreign genes into target plant cells and tissues. The foreign gene is cloned in the tDNA region of TA plasmid in place of unwanted sequences. In animal, there are four general classes of gene transfer methodology that can be applied to culture animal cells, but transduction and transfection are the most widely used. Transduction is a virus mediated gene transfer in which the foreign DNA is packaged inside a virus particle. Transfection involves the use of chemical or physical methods to persuade cells to take up DNA from the culture medium. There are numerous different transfection methods: chemical transfection, liposome mediated transfection, lipofaction, electroporation, receptor mediated endocytosis. Then direct gene transfer method involves the direct physical introduction of DNA into the cell. The common example is microinjection. Microinjection is usually applied to animal eggs, zygotes or early embryos. Particle bombardment, which involves the acceleration of high velocity DNA coated microprojectiles into cells, is another direct transfer method. Next is about polymerase chain reaction which we will be learning with the help of next video. Thank you.